Welcome back to the deep dive. Today we're looking at a stack of sources that, I think, describe a fundamental pivot in structural biology.
Yeah, I'd agree with that.
For years, the whole conversation around AI and molecules, it's been about prediction.
Yeah,
Trying to figure out what a structure is.
that's right. Tools like AlphaFold were incredible, absolutely revolutionary. They gave us that static picture. But that was always just half the story.
So what's the other half?
The new frontier, the one we're diving into today, is all about moving from that prediction to programmable design
Mm-hmm.
and dynamic simulation.
So not just looking at the molecular world, but actually, controlling it?
Exactly. Our sources today lay out a kind of unified molecular operating system. We're looking at three huge breakthroughs that all work together.
Okay, let's lay them out. First up is a model called ODesign.
Right, a generative world model that designs brand new functional biomolecules completely from scratch,
Then there's GraphFormer.
which leaps past those static snapshots to predict the full functional movement of these molecules, their equilibrium dynamics.
And finally, because these new tools create so much data, we need a way to check their work.
And that's where eRMSF comes in. It's a robust, unified analysis tool.
So our mission today is to unpack how this whole stack, this autonomous molecular intelligence, gives us a level of control that gets us closer than ever to programming biology like it's software.
Let's do it. And I think we have to start with ODesign because that model really changes the definition of what molecular design even is.
Okay, so ODesign. It's being called the first of its kind generative world model. That phrase alone is huge.
It is.
But what really grabs you is the scope. It supports all-to-all biomolecular interaction design.
And that all-to-all part is the key. We're talking about one framework that can design connections between proteins, nucleic acids, so DNA, and RNA small molecules, and even ions.
The whole biological toolkit in one program.
Yes, and that unification is the real technical marvel. You have to remember, historically, these fields were siloed. If you were a chemist, you designed a small molecule with atomic rules.
Right, and if you were a biologist, you were thinking in terms of protein residues.
Exactly. Two totally different languages, two different sets of building blocks.
It's like a CAD program trying to handle architectural blueprints and quantum physics diagrams at the same time.
That's a great analogy. ODesign breaks down that barrier. It does this by abstracting all those different chemical building blocks into a single unified token space.
It creates a universal language.
It learns the universal language of physicochemical interaction across everything. And once it knows the rules, it can generate novel structures using any combination of them.
The level of control here seems revolutionary. We're not just saying design a protein that binds here.
No. You can specify conditions down to the precise atomic level, the motif level. It gives the designer molecular torsors.
And it has two design modes.
It does. There's the standard rigid receptor mode where your target is fixed. But the really innovative one is the flexible receptor mode.
What does that do?
It lets the model co-generate both the target and the binder together. It understands that receptors shift and adapt in nature, so it adapts the receptor's shape while it's designing the perfect partner molecule.
Okay, let's get to the practical payoff, because this is where the speed and efficiency numbers are just jaw-dropping. This has to be the aha moment for any R&D lab.
Oh, absolutely. So in a benchmark for protein-binding protein design, ODesign came up with an average of 2,672 successful designs
2,672.
per target in just 24 hours on one GPU.
Wow. How does that compare?
Well, the second best method, RF diffusion, it averaged only 555 in the same time.
So that's an order of magnitude leap.
It's a massive leap. This isn't a minor tweak. It fundamentally changes the economics of molecular research. You're cutting out months, maybe years of screening time.
And for the really high precision stuff, like designing enzymes.
Even more dramatic. For atomic motif scaffolding, which you need for that kind of bespoke enzyme design, ODesign showed a 37.5-fold efficiency improvement.
37.5.
That means a single computational run can generate enough high-quality candidates to fill an entire 96-well screening plate. That used to take a team weeks. Now the bottleneck is no longer the design, it's the physical screening.
And because it's this unified framework, it's opening up design spaces that were, too complex before or didn't have enough data.
Exactly. Look at nucleic acid aptamers.
These are synthetic DNA or RNA molecules that act like antibodies, right?
That's the idea. ODesign successfully designed protein-binding RNA aptamers in a zero-shot scenario, meaning it just figured it out without specific prior examples and got a success rate of almost 78%.
And the sources mention it also work for DNA aptamers. That feels important.
It's crucial. RNA aptamers get studied computationally a lot, but DNA versions are way more stable, more resistant to breaking down, which makes them much better candidates for actual drugs.
So what's the most challenging area it's tackling?
I'd say it's nucleic acid binding small molecules. The main database for this, PDB bind, shows that less than 1% of known complexes involve a nucleic acid binding to a small molecule.
So there's almost no data to learn from. A tiny haystack with very few needles.
Precisely. And ODesign is one of the very first models to successfully navigate that. It's generating brand new strategies for drugs that target DNA, like some anti-cancer agents, or RNA, like certain antibiotics. It's finding starting points where humans were just stuck.
So, okay, ODesign gives us the perfect blueprint, the perfect static image. But as you said earlier, life is dynamic. A molecule is only functional if it moves correctly.
Right.
So if ODesign builds the perfect key, DGraphFormer has to show us if the lock will actually turn. Is that the idea?
That is the critical transition, yes. The big limitation of predicting a single structure is you often miss the functional story. Proteins have to switch between states to work.
They're not just sitting there.
Not at all. Say a protein needs to switch between a closed state A and an open state B. A static model might just give you some weird, unstable structure, see somewhere in the middle. It gets the shape wrong because it misses the behavior.
A fundamental disconnect between structure and function.
It is. Take BRAF kinase. It's vital for cell signaling. It is an active, open state and an inactive, closed one. Single prediction models often just fail to see those two distinct, stable states.
And if you're designing a drug to lock it into one of those states, you need to know the whole map of its movement.
You need the whole map. And this idea of the entire equilibrium distribution can even explain things like viral infectivity.
The Omicron spike protein is the big example here, right?
A prime example. Its increased infectivity wasn't caused by a huge change in its most stable shape. It was a subtle shift in the distribution of its three available states. If you only looked at the single most common structure, you would completely miss why it was so successful.
Okay, so traditionally, mapping that whole distribution required molecular dynamics, or MD. Why is MD so slow?
It's just inherent to the math. MD relies on statistically dependent sampling. Every new step depends on the last one. So to get from one stable state, a valley, to another, the simulation has to cross these huge energy barriers.
These are the rare events?
That's them. The simulation literally gets stuck. It just sits there, waiting for some random thermal bump to push it over the barrier. It's like rolling dice a million times just to get over one hill.
And every roll depends on the last one.
Exactly. DGraphFormer sidesteps that entire problem. It uses generative AI, a diffusion model, to do what's called IID sampling.
Independent and identically distributed.
Right. Doesn't follow a path. It just generates structures independently all over the map, allowing it to recover the whole distribution almost simultaneously.
We need to pause here for the metric that really puts this in perspective.
The big one.
For a SARS-CoV-2 protein simulation, getting the full equilibrium distribution with traditional MD took 2.6 milliseconds of simulation time.
And that 2.6 milliseconds was achieved by the folding at home network, which took about 70 GPU years of computational effort.
70 years of processing time. So DGraphFormer.
DGraphFormer, using IID sampling, recovered 70% of that same distribution in just 18 GPU days.
That's not an optimization. That is a paradigm shift.
When you go from 70 years to 18 days, you've gone from a problem you solve once in a generation to one you can solve routinely in a lab.
I have to ask, though, 70% of the distribution, what about the other 30%? Does IID sampling have its own biases that MD, for all its slowness, avoids?
That is the crucial question. And yes, it can. MD is, by definition, kinetically accurate. It follows the physical path. Generative models can sometimes oversample areas they were trained heavily on, or maybe undersample some really obscure states.
Which is why you need the next tool.
Exactly, why you need tools like eRMSF to check for and quantify those potential biases. But for many applications, the trade-off is more than acceptable, especially given what else DGraphFormer can do.
Like calculating energy.
Yes. It's not just sampling shapes. DGraphFormer can estimate the exact density of the distribution. And you need that density to calculate the microscopic properties that really matter for drug discovery, things like free energy and entropy.
So you can pre-screen how well a drug will bind before you even make it in a lab?
That's the holy grail. And it closes the loop with property-guided inverse design. You can tell the model, show me only carbon structures that have this specific band gap property. You're designing for function from the very beginning.
Okay. So we have design from ODesign, we have the dynamic function from GraphFormer, but the data is coming from all these different sources. The 70-year MD run, the 18-day GraphFormer run.
Other AI models too?
We need to compare apples and oranges. And that brings us to the validation tool, eRMSF.
Precisely. You can't use traditional metrics on these different kinds of data.
Right.
You need a unified tool to compare their predicted dynamics fairly.
Let's define the terms. The classic concept is RMSF, root mean square fluctuation. How does eRMSF generalize that?
So traditional RMSF measures how much an atom wiggles over time in a simulation. But AI models often produce a static ensemble, a collection of snapshots with no time order.
Right, no timeline.
So eRMSF, or ensemble-based RMSF, generalizes the idea. It measures the positional variability across any set of structures relative to a reference, which gives you a consistent baseline no matter how the data was generated.
And the key is the segmented analysis, which it shows as a heat map.
That's where the deep insights come from. Take their example of AIDK, a protein that goes from closed to open. Standard RMSF just tells you which parts are flexible. But eRMSF, with its segmented approach, could actually localize that big transition to after about 40% of the trajectory had passed.
Hang on, so it adds a time-resolved dimension back into the analysis by chunking the ensemble into early and late stages of the transition.
That's the power of it. You're getting a kinetic insight from a structural analysis.
Yeah.
It's incredibly valuable for understanding the actual mechanism of how a protein changes shape.
And finally, eRMSF is what exposes the biases we talked about earlier.
Yes. This is where you see the proof. When they paired ensembles of another protein, Abl kinase, eRMSF showed that alpha-fold subsampling captured very restricted movements around 2 angstroms.
It stuck close to its first guess.
Pretty much. The MD simulation showed more fluctuation up to 4 angstroms. But another AI tool, BioEmu, showed the highest flexibility of all. It was sampling a much wider space.
So eRMSF is the essential interpreter. It tells you exactly what kind of answer you're getting from which tool, so scientists can pick the right one for the job.
It provides the quantitative framework you need to have confidence in a field that's just being flooded with new data. Without a unified validator like this, the breakthroughs in design and dynamics would lack credibility.
This has been absolutely foundational. We've gone from just predicting structures to this unified modular system. ODesign makes the blueprint.
For any biomolecular combination.
GraphFormer predicts the full dynamic function and the energy landscape.
And eRMSF unifies the validation and checks for bias across all platforms.
It's the entire molecular pipeline driven by AI.
And this modularity, as the ODesign developers themselves point out, is hinting at a much bigger future. The next logical step is to evolve ODesign into a self-evolving molecular world model.
A closed loop.
A completely closed loop. Imagine a system that can improve itself through in silico evolution. It designs a molecule, simulates its function with GraphFormer, evaluates the result with eRMSF, and then uses an LLM-based reasoning agent to figure out how to improve the design for the next cycle.
That's not design assistance anymore. That's the groundwork for autonomous molecular intelligence.
And that's the thought to leave you with. If these systems can design, simulate, and analyze the function of basically any combination of biomolecules, the challenge shifts. The new question becomes, what complex biological problems, what currently unsolvable diseases or unprecedented materials will they autonomously solve next?
Thank you for joining us for this deep dive into the world of autonomous molecular intelligence. We'll see you next time.